Hello friends, welcome to yet another sneak peek into our biology and bioengineering summer camp. In the summer camp, you will learn about the world of biology and bioengineering in the 21st century. Today, let us meet our guest speaker, Rutik Thorat, who will join us at the summer camp. Rutik is currently a scientist with HiMedia Labs, Mumbai. Rutik, an Indian researcher working with HiMedia Labs in Mumbai, is known for his award-winning research on sickle cell anemia. Rutik himself suffers from the disease. Driven by a desire to know more about the disease that kept him sick, Rutik took up the challenge to design drugs for improving the lives of people suffering from sickle cell anemia. Rutik, at a very young age of 19, has come up with the novel peptide-based drug therapy for sickle cell anemia that prolongs the quality of lives of sickle cell anemia patients. Currently, he is also working on treatments to combat cancer and COVID-19. Rutik has represented India at the ISEF, the world's biggest science fair conducted in Phoenix, Arizona, United States. He has been featured in Forbes India magazine and several other media platforms. We are all excited to have Rutik with us. Let us listen to what he has to say about the summer camp.
All right, everyone. So Rutik is already here with us. So Rutik, welcome to the summer camp. And
yeah, thanks for having me. Yeah, everybody hi.
So please you can share your screen, and if you have a presentation,
Yeah,
you
sure.
can start.
I'll just.
Your slide is good to go.
Yeah. Okay. Everybody. Hi. I'm Rutik. So I started researching about sickle cell anemia at the age of 16, and I actually came up with a therapy for sickle cell anemia, a novel peptide-based drug therapy for sickle cell anemia. So basically, what I did was, so basically in sickle cell disease, you have normal blood cells. Just allow me a minute. I'll just connect a laser point so you can get, Okay. So what is sickle cell disease? I'll tell you about that. So as you can see over here, these are your normal cells, which are normal red blood cells. But in sickle cell disease, due to genetic mutation, it turns into a crescent moon shape or banana shape, you can say. So these cells are usually flexible. Your RBCs are usually flexible. So they can just fold and squeeze it into your small blood vessels. But the problem with sickle cell anemia, due to your genetic mutation, this crescent moon shape cells have difficulties in traveling through your small blood vessels. And with that, you have, many other difficulties too. So basically, what happens is like this blood vessels flow through your small blood vessels, and then they get stuck at one point, and the cells that are coming from behind, they also get clogged up and they form a blood clot, and which leads to reduction of your RBCs. So this is the major symptom that occurs in sickle cell anemia, which results in pain, and then your muscles are weak, and then your bones are weak, and so on. So basically, sickle cell anemia is mostly found in Indian, Middle East, African cities, and now you can find it everywhere. But this was majorly initiated from African cities and India and Middle East. So Yeah. So what happens is like your hemoglobin, which is your essential part of your RBC, is it is abnormal. So it is given the name as hemoglobin S, which state the hemoglobin of sickle cell anemia. So it lacks the affinity to carry oxygen that much of a compared to your normal RBCs. It lacks the capacity to carry the oxygen. So it is really difficult to when the cells cannot carry oxygen to your other body organs. I mean your tissues or your essential organs. So which is a major problem in that. So oxygen deficiency is one of the major problems. And then there is health burden, because this is kind of a rare disease, and despite of available therapies, your lifespan is shortened by 2-3 decades. So I thought, as I was suffering from sickle cell anemia myself, and I thought at the age of 11, I started reading about this. And fortunately, I mean, I acquired the knowledge about this field by talking to the doctors and all. And there was an unfortunate event in my life. I lost my father due to the same disease, so which really boosted me up to take this as a research. And at the age of 16, I started mailing a couple of scientists, asking them that I want to work on certain therapy that I have designed. Will you allow me to work in your lab and all? I mean, there were a couple of rejections, the criticism, and then you get good scientists. So I got connected with Dr. Sanjeeva, and then I went to proteomics lab, and I learned proteomics and little about brain tumors, glioma, and it was a enriching experience. And after that, I took some of that basic biology experience and advanced biology experience to some other scientists to help me work on sickle cell anemia. So I got connected with a company called HiMedia, and then I started my research over there. So what I suggested was a immunomodulatory and anti-inflammatory drug. So this drug RS has a pleiotropic effect. So which is it gives, the one drug gives multiple advantages. So what was the main criteria to see in that? The drug was a disease modifying treatment for sickle cell anemia. So the approach was simple, that there is inflammation caused in sickle cell anemia. This therapy acts against the inflammation. There is the patients are immunocompromised, so which means that they are prone to any infection, any kind of infection. So what I did was I took that in mind, that this drug can act on immunity also. So this drug actually acts on immunity too. So as it is an immunomodulator, and then it has multiple advantages apart from that, like angiogenesis, which is like rapid growth of your blood vessels, and then there is fibrinolysis, and there is anti-apoptotic property, and so on. I mean, so the main purpose was to see that it enhances antioxidant activity, which is like reducing your ROS species, which is reactive oxygen species, which damages your DNA, lipids, and the essential parts of your cell component. So so I just took this hypothesis and we started the work. So what we did was in vitro development plan. So in lab, we took a cell line called GM209020, so which is a sickle cell anemia B lymphocyte cell line. So what is cell line is basically you take cells and grow it in artificial conditions, so in lab. So any drug, the process is like, first you go from in lab, then you go to animals, and then you go to human patients. So that is which are known as clinical trial. And I'm sure that previous lecture you got an idea of what are clinical trials. So basically, I designed experiments. So the first state is like you have to design your standardization of dose, like what dose will go into your body and at what time, like till what time its effect would be there. That you have to take into consideration. So what I did was like we took I get 4 dosage from 0.1 nanogram to 1000 nanogram, and we distributed the time to like 0 hours, the effect seen in 0 hours, then 24 hours, then 48 hours, and then 72 hours. And that was one experiment that we did. Then the flow cytometer, then Eliza test. So Eliza is like your immunoassay. So you see the immunoglobulin bodies that have been produced in the body, like at what level it has been produced. So then we did antioxidant enzyme estimations. As I told you that the species which kill ROS species, like reduce ROS species that are antioxidant enzymes. Then we did the long-term effectiveness of the drug. Like once the drug is given, a single dosage can last up to how many days or how many weeks that we wanted to see. So yeah, let's begin with this. So basically, as you can see, this is the first chart. So as I told you, the standardization of drug. So as you can see over here, we took 4 time slots, which is 0 hours, 24 hours, 48 hours, and 72 hours. And these were the these were the drug amounts that were taken. So 0 nanogram, which is like control. So there's no drug in it. Then you have 0.1 nanogram, 1 nanogram, 10 nanogram, 100, and 1000 nanogram. So what we were seeing was the cells that have been dividing at with the effect of the drug. So as you can see, the highest number is over here. So which is 100 nanogram at 48 hours. So this graph was showing a significant amount. So we got our dosage, which was 100 nanograms, and the time, which was 48 hours. So the cells were dividing majorly in this. The proliferation was seen majorly in this. So that was the test that we did. Then we did CD19 surface marker test. So which it is an antigen test. So you see the immunity of your B cells. So the expression of CD19 was seen in this cell. So we took 4 combinations. Now the drug experiments really start. So this 4 combination throughout the lecture you will see everywhere. So these are fixed combinations. This blue color, as you can see over here, these are cells, just the cells, B lymphocyte, sickle cell anemia B lymphocyte cells. Then we took LPS. So what is LPS? Is lipopolysaccharide, which is an infective agent that we took because, as I told you earlier, that these patients are immunocompromised. So we wanted to see that what if any infection happens, then what happens to the cell? And with the effect of drug, what happens to the cell? Then just the cells and the drug. So normally anything is not happening. We are just taking the drug to treat the symptoms. And then the 4th combination was like cells with your infection and with the drug. Like what happens when you take the drug while you're having an infection. So as you can see the results, here you are having a significant value, which is 0.002. So there is higher expression of CD19 with cells plus LPS plus your drug, RS drug. So which shows that the drug is increasing CD19 expression, which is a good thing for enhancing your immunity. So yeah, then we move to Eliza. So basically Eliza is like IgM production in your body. To see IgM secretion, we used to see like we use the test to see IgM secretion. So basically, whenever an infection affects your body, these are the parameters like IgM or IgG bodies that have been seen to see the immunity of your body, like at what level it fights back. So, so these are extremely, as you can see over here in cells, it is already low, the expression of this thing. And with the cells plus LPS, when the infection occurs, the IgM expression has gone little up because basically it is a normal, defense mechanism activated in your body. So your body is fighting for it. And as you can see with cells and drug, it has been a little bit higher. The IgM production has been enhanced as compared to your normal cells. And when the infection occurs and then you take the drug, the expression has been enhanced significantly. So we got one more check over here, so that the IgM has been increasing, the immunity is increasing again. Then we come to the next experiment. So now I wanted to see that I wanted to compare a normal human being with respect to sickle cell anemia, that what this drug is really doing. So I took Dowdy cells, which are normal B lymphocytes, like normal human B lymphocytes, and compared it with sickle B lymphocytes. So basically here you can see these are sickle B lymphocyte on the first chart all here, and Dowdy cells, which are normal B lymphocytes. And what I was seeing is like proliferation in specific Rs. So like cell proliferation is basically dividing of the cells, and your cells are dividing uniformly, and there's a good growth to fight back any infection and all. So here you can see that again we took the same combinations throughout the experiment. Cell, it just was the cell. So here you can see significant growth in this SCA B lymphocyte when it has been given with when it is infected, because your cells are fighting back as normal defense mechanism. And when the drug is given, it is significantly seen that it is controlling the condition as compared to this normal cells. So that was one big thing. So now we move to antioxidant activity, which is a main showstopper, because the antioxidant activity in sickle cell anemia is very low because of the immunity is not good, and there are other factors also that are affecting that. So here, same as you see that this is significantly increased. The cells with when treated with drug, it is increasing your antioxidant activity, and which shows that the drug is strongly enhancing the activity as compared to your normal when your cell is infected with the cells, or any other parameter you can see the normal cells, the antioxidant activity is low over here, but when you're treating with RS drug, it is significantly high. So yeah, so basically that showed us that the cells, the B lymphocyte, can fight against the antioxidant oxidative stress. So it is, and it was again showing a protective action of B lymphocyte. And yeah, furthermore, we did this long-term effectiveness. So what you do is like you take drug once. Now at day one you have taken the drug. Now we have to see that till how much days its efficacy is valid. So we gave the drug and we saw that till day one dosage was valid till 7 days. So which was a good thing. So like once you take the drug, you have it till 7 days, and then in humans it would be varying because it can be more, because we are taking a small amount of cells. So what we did was we saw this capacity by measuring the cell death. So how many cells dies? With respect to that, we saw the test. So basically, when you take just SCAB lymphocyte cells, there is huge cell death. As I told you, when the cells get stuck in your small blood vessels, it occurs a blood clot, and which leads to hemolysis and destruction of other blood cells too. So as you can see, the cell death is higher over here by just the SCAB lymphocyte. But when an infection gets in the picture, your cells are dying more because you're immunocompromised and you're at very weak state. So this is a big thing. I mean, the cell death is higher. But when you treat with our drug, so here there is just normal cells, you can see, and see with the drug, the death is rapidly decreasing over here. So which shows that our drug like enhances the lifespan of the cell. So which is a big big marker to see over here. Then if an infection occurs and then we give the drug, you can measure this significant gap, and you will get to know that with the drug your cells are maintained and your death has been reduced. So this were the checkers that your cells, the drug is working well. I took this kind of main aspects of the cell, and then I just measured it. So to just give you a summary, first we did the IgM test, Eliza one. Without the treatment, it is very bad. With the existing drug, it shows no effect. And with the treatment of RS drug, it is happier. And then accordingly, you can see overall performance. So without the treatment, patients usually die, or it is difficult to maintain in the condition. With the existing drug, there are again very frequently occurring symptoms, and the existing drug cannot cure it. It is just an management of symptoms, which has side effects, a lot of side effects, and it is kind of difficult to manage it. So with RS, you're getting a good immunity power, and with other effects too. So yeah, so that was a summary of that. And then I made this is B cell, and this is drug molecule, you can see over here. So IgM has been increased, and your drug is eating up the reactive oxygen species, which are providing damage to your DNA lipids and eating up the oxygen, as you can see. So yeah, pretty much all. Yeah, so that was from my side. Now,
All right, Rutik. So I think, excellent work done. And
yeah,
we can take one or two quick questions from the participants.
yeah, sure.
I think there's some question in the chat from Mahima Shukla.
Oh, I'll just.
How you design RS drug?
So RS is actually a code name given to the drug. So I cannot disclose the name. It is an immunomodulator drug. So it was a recombinant protein. I can say that that I took into consideration, and then I worked on it. So it is like it was studying the effects of the drug, and then I just applied. I just saw the unmet needs of this sickle cell anemia disease, and just matched with it. And then you get to know that this is working. So yeah. Any more questions?
Yeah, no, so Rutik, you have set up really, great example of how to fight
Yeah.
against the disease rather than, thinking to give up. Why not work, closely on that and try to understand the disease and try to, come up with the potential ways of therapeutics, right? So wonderful.
Yeah,
Just
thanks a lot.
to ask whether Dr. Om Srivastava is in the audience right now.
Yeah.
Dr. Om Srivastava, are you there already joined or not yet? So we can take one or two questions, Rutik, in the meantime. So Ojasvi, if you want to ask a question.
Hello, Rutik. Actually, your focus was amazing. I am just enjoyed your lecture today. So,
Yeah.
like your like doing sickle cell anemia, this peptide therapy, like it would be possible for chromosomal disorders like Down syndrome, bubble boy syndrome. There are a lot of things, right? So it would be possible to do
Yeah.
with this like how do we manage this things like you done in sickle cell anemia? So could you tell me what are the process like how do you went through this process? So just a queries to ask that. Yeah, thank you.
So basically, you need to have interest in a specific field or like any subject you take up, and then you go a year through study, or I would say 2 to 3 months studying the literature of it and seeing the possibilities that where you can work on. So as you know, people have said about cancer. So there are many undiscovered fields. I am sure that sir have told you many undiscovered fields are there. So you take that point, and then by studying literature you can match anything like which is already working, and you can apply that thing that any specific thing to into implementation, that if this thing is working with certain disease or not. So yeah, basically it is a trial and error, I would say. But yeah, you need a lot of study of literature background, and yeah, I mean with interest you can achieve anything. I mean, yeah, that
In
would
this
be the
case, I
short.
think Rutik made a good literature review to really come up with a hypothesis, and then that helped him to plan his experiments, right? So, Sharanya, you want to ask
Yeah.
any question?
Hello, I'm Purvaja Ganesh. Can you tell me what makes the red blood cells become like a crescent?
So it is actually a genetic mutation in your there is genetic mutation in your beta hemoglobin. So there is a specific position in your chromosomes. Like suppose you are in sickle cell anemia specifically, it is in occurs in 11th chromosome, wherein your glutamate changed to valine, and so valine is a hydrophobic amino acid, and which this small defect of changing one amino acid to other can cause a bigger problem. So there are now you can ask that there are multiple therapies for sickle cell anemia that are coming up, such as gene therapy, which is targeting this specific thing, specific chromosomal site with the help of CRISPR, and then there are other also like other drugs also. So even I am working on that too. Apart from that, I'm even working on COVID and cancer therapies. So yeah, that is pretty much it. I mean, due to this small amino acid change, base pair change, that this bigger problem occurs. So yeah.
All right, good. So,
I hope that cleared up your drum.
Dr. Om, have you joined now?
Yeah, I got it.
So we'll just take one last question before we take the next session. So, Ojasvi Singh, you want to ask a question from Rutik?
Yeah, sorry, my internet connection is unstable. I'll just. Okay. Yeah, am I audible? Yeah, my connection is unstable a little bit. Yeah. So basically, if you have any doubts, you can reach to me. I'll just share my mail in the mailbox. Or just a minute, I'll just share it over here. Okay, so this is my mail. You can just reach me out on any of the platforms. And yeah, thank you for your time. I mean, I look forward that one of you can come up with a good project and literally change the world. Yeah, I guess we can take up last out, right?
All right.
Any questions?
So Rutik, yeah. So I think your internet was slightly weak, and the voice was breaking, but I could get a sense of it. So definitely,
Yeah.
I think good opportunity for all the students here to think about, irrespective of your age, you can always come up with the new ideas, and then plan to come up with a hypothesis, the experiments, and then see how
Yeah.
that can take you forward, right? So with that, let me thank Rutik for the wonderful work
Yeah.
he's doing and a very good lecture he gave today. So thank you, Rutik.
Yeah, thanks a lot. Thanks for having me, sir. And yeah, everybody bye.
Great. All right. So next will be the last